Right, clear? Are you able to see graph?
Yes, sir. Yes, sir.
Right, right, right. So let me complete here. The first, the single stranded DNA percentage gradually decreasing, and the completely when reach the bottom, the completely all single stranded DNA get reassociated. Right? In this process, first, as I said, high repetitive DNA reassociated, next moderative reassociated, next low repetitive reassociated. The finally the single stranded DNA becomes double strand. After reassociated, this single stranded separated DNA get reassociated or get renatured. Right? This is cat value, and this is mainly observed in prokaryotic, especially in bacteria. If the same experiment carried out using the DNA purified from the complex eukaryotes, for example, human beings, then the simple sigmoid curve is not produced. This is sigmoid curve. The S-shaped curve called as sigmoid curve. The sigmoid curve only produced in the bacteria, single cell bacteria. But if the same experiment, if we conducted when we taken the human genome copies, single cell DNA copies, we could not expect this type of sigmoid curve. Instead of a curve produced that is the sum of the re-annealing many different components, but has three main plateaus. These present three main classes of DNA molecule. So anyway, finally, this sigmoid curve mainly occurred in DNA. So this is cat value or kinetic of DNA reassociation. This is important for short answer, right? And the next in page number 16, 16, the given the other important aspect of genes, genomes, the family, pseudogenomes, they are again repeated. That's why uh, now directly enter into the third unit, that is page number 61. These topics repeated several times. That's why I would like to tell only one time, that is page number 61, eukaryotic gene structure at DNA level. Okay. And next third unit and these are all the repeated DNA genome sequence, chloroplast and mitochondrial genome, and again genome organization. Genome organization repeatedly occurs in the genome organization. Third unit, 4.3, organization of eukaryotic chromosome. Already we have discussed yesterday the same topic, right? So here the most important topic is eukaryotic gene structure. Eukaryotic gene structure at DNA level. This is important, very important for further lessons. And further lessons, that is gene regulation for gene regulation, and also gene expression. If you know about gene expression in eukaryote and gene regulation in eukaryote, you should know about the gene structure. Let's see the gene structure at DNA level. Right. Are you able to see whiteboard? Hello? Whiteboard. Hello? Voice is clear or not? Hello? Don't draw, don't draw. My voice is clear or not? Are you able to see whiteboard? Hello? Hello?
Yes, sir.
Are you able to see whiteboard?
Yes, sir.
Right. Now, another important topic, eukaryotic gene structure at DNA level. If you take the eukaryotic gene, this is eukaryotic gene for DNA, gene structure, DNA level. Right. The first of all, in gene structure, we should know about the exons, exons, introns, exons and introns. And another important part is, let me change color, promoter, promoter gene, promoter site, promoter, promoter.
Sir,
Silencer
unable
and
to see
enhancer.
the
What?
unable
Hello?
to see.
Whiteboard not clear? Hello?
Whiteboard is clear, sir, but I am unable to see structure, sir.
No, no, no. A marker. Are you able to see the marker writing? Marker writing. Hello? Are you able to see what I am writing on the board?
Yes, sir.
Promoter exons, red color gene, red color stands, in red color stands, exon, intron, and another gene part is promoter. All are visible or not?
Visible, sir.
Right. And apart from the promoters and enhancer and silencer, also part of the gene, enhancer, enhancers, and silencer. We will discuss later about enhancer and silencer. But here the most important in gene structures, introns and exons. Introns and exons, they are the part of the gene. If the gene, it should consist coding part and non-coding part. In this, the exons are coding. Exons are coding part and introns are non-coding part. In protein synthesis, protein formation, the only the exon active involved in the preparation of protein strand, but not introns. Of course, the introns also part of the gene, but it's not code for any amino acid, amino acids and any proteins, right? And another important reason, the UTR, that means untranslated region. UTR, untranslated region, untranslated region, 5 and untranslated region, 5 and untranslated region, 3 and untranslated region, 3 and these also part of the gene, these are part of the gene structure. So apart from the exons, introns, promoters, enhancer, silencer, and trans. Here also untranslated region present. A typical multi-exon gene as following the structure. It start with promoter gene. So this exon here, this exon, the start with promoter gene here. This is promoter. Here promoter, I will explain clearly about the promoter, what is the concept of promoter, and which is followed by the transcribed but non-coding region, which is called phi and translated region. Now the first promoter, after promoter immediately untranscribed F1 region present, F1 region present, but it code for any amino, not code for any amino acid, and this follows the again in initial exons. This is initial exons. Now this is initial exons. Now the initial exons, it has the start codon. Start codon. Following the initial exon, there is an alternating series of the introns and internal exons. Now the exon, it is start codon. Start codon means it is synthesizing the strand. It is involved synthesizing the strand. And later again introns, not code for any amino acid, not code for any base. And again its continuation, the strand continuation till until it reach the untranscribed region 3 end. Untranscribed region 3 end. In 3 end also it's not transcribed. That's why end the preparation of strand. And this is clearly understand in transcription process. I will explain later about transcription eukaryote. So just bus this topic concept is the what are the gene part, what are the gene structure, and what mainly involved in transcription process, but it is not entered into the transcription. We are discussing only gene structure. First of all, in gene structure, what is gene? Hello, first, what is gene? Gene. Hello, what is gene? What is DNA? Am I audible? The simple genes are made up of sequence of DNA and basic unit of hereditary, and which is passed by one generation to the another, from parent to child. Hello? Hello?
Yes, sir.
Am I clear? Voice clear or not?
Yes, sir.
Any disturbance from my side?
No,
Please
sir.
inform me. Please inform
No,
me.
sir. No, no.
Yesterday also I have seen my class in YouTube. When I see some of the marker spotters different, not exactly posted when I teach the class. Please inform me. Please inform me the type of the mistakes. But when I am explaining, but I will show. I am saying these are very clear. Right. So far clear or any disturbance? Hello? Yesterday when I explained the classes, actually I have explained it based on the diagram part. The diagram fit somewhere and marked somewhere. When I observed in the YouTube classes, but I clearly explained with help of the diagram. But there is no matches when I observed in YouTube class. If any such type of mistake, inform me. While you listening classes, please find out that mistakes, any errors. So far, any disturbance? Hello? Salaiha, Nagarani, Navadeep.
No, sir.
Are you clear?
Yes, sir.
Right. Right, in gene structures, exons, introns, promoter, enhancer, right. These are the sequence. First, I will explain later about enhancer or silencer. After that, which follows the promoter region, and from after promoter, the untranscribed region, 5 hand. Now, which follows the exons. Actually, this is the active site exons, which codes for amino acids, and this is involved for formation of the stand, mRNA stand during transcription process. And now and there, the introns also present, but they are not transcribed for anyone, any amino acid. And finally, end with untranscribed region 3 hand. Untranscribed region 3 hand. These are all part of the gene. They are part of the gene, right? Now, Alda told you the gene definition. What is gene? Genes are made up of sequence of the DNA, and this is basic unit of heredity passed from the parent to child. Now clear with the DNA and gene. Now next one, the another important part of the eukaryotic gene is alternative start and stop producing related proteins. Alternative start and stop producing. For example, here alternative start and stop related. Two proteins can be in this. Two proteins. Two proteins can be generated from single gene. That means, for example, this is the gene. This is the gene, right? So as I said, mm, UTR untranscribed 5 and exon. Actually, exon is the start point. This is actually plus start point. From start point this side downstream, these also important. And this minus side upstream. In upstream, UTR present, untranscribed region 5 and present, and also PR promoter present, and promoter regulated by enhancer, silencer. Silencer or enhancers. I will explain later about this. Right. So anyway, from the start point, from the start plus point, this from the start point, this actually start point, and on the starting point, let's change color. mRNA stand prepared, mRNA stand produced, transcription process. But somewhere here, UTR 3 and present. For example, here the UTR 3 and present. UTR 3 and present. Now this transcription process continuation until the 3 end, and one DNA strands formation. Later again start from this side after 3 end stop codon. Again this transcription start. Again transcription start, and it forms the another mRNA strand from this side. But this one shows continuation. The starting one shows continuation. This mRNA strand shows continuation. This first mRNA, sorry, this mRNA strand shows continuation. Now, how many genes in this? How many genes here? How many genes? How many protein strand? That means actually this is mRNA and blue color also mRNA, and these two mRNA prepare the proteins. Proteins later on the mRNA strand protein formed that okay directly this is the protein strand, and this protein synthesized from the hello from where this protein synthesized,
Exon.
right? Exon part of the gene, right? And next another blue color protein synthesized from, but the taking consideration this entire red color this box, let this way, actually this box is the gene. Remember this box is gene, but in this genes are coding and non-coding all are present, all are present. But now start with this first exon series, first exon series, first exon mRNA prepare the stand. But while the preparing transcription process, the somewhere UTR untranscribed 3 region present. When untranscribed transcription region present, that the end the transcription process and again start, but again start with the blue color protein stand, blue color protein stand, right? Now, first of all, let me give answer one thing. How many genes here? How many genes involved in this? How many genes involved in this structure?
1.
Only one gene. But the one gene importance is alternatively start and stop. Alternatively start and stop. And start and stop. That's why how many proteins are formed on the single gene?
2.
Two proteins are formed. Two proteins are formed on the single gene, right? So this is also one of the part of the genome, one of the part of the gene. So apart from the exon and entrance, and apart from the UTR untransferred region, apart from the promoter, and some of the gene parts are the alternately start and stop producing related proteins are present. These proteins involved in formation of from the one gene, one single gene to different proteins. So this is the one part. Another somewhere overlapping genes also observed. Overlapping genes. Overlapping genes also observed in eukaryotic genome. How the overlapping genes are formed. Here, another one, the two genes may share the same sequence. Now, in this, genes are two, but the proteins one. The two genes may be share the same sequence by reading frame. The two genes overlap when the same sequence of DNA shared between the two homologous protein. Let's see, for example, here the first of all, for example, this is the gene sequence A, T, G, C, A, thymine, guanine, cytosine, thymine, guanine, guanine, thymine, cytosine, guanine, guanine, adenine, guanine, cytosine, like this. Now overlapping genes here A, T, G, C, T, G, G, T, C, G, A, guanine, cytosine, thymine, thymine, guanine, summer. Now here if you take the triplet 3, 3, 3 triplet nature. Now this is ATG, CAA, ATG. And again if you take this ATG, this is triplide. ATG triplide. But here the ATG triplide. This is the overlapped gene. Overlapped gene. But if you observe the when the code formation, these three code for one amino acid, code for one amino acid, and these three CAA code for one amino acid, and TGC these three TGC code for another amino acid. Like this, every three triplet codons code for amino acid. But what happened here? This ATG, the overlapping another genes, ATG genes. And here also CTG, cytosine, thymine, guanine, cytosine, thymine, guanine, the same cytosine, thymine, guanine. But the triplet is different. Triplet is different. For example, if we take the second triplet codon, CAA, one triplet codon, code for one another of this. Then overlapped genes ATG, one triplet codon. But in above, this ATG not a triplet codon. In this triplet codon is T, thymine, guanine, cytosine. Are you observed? Hello? Are you able to see?
Yes, sir.
Right. Please careful. Please careful. Right. So here what I'm saying, the first, this is my marker. This one, let's change the color. The blue color one gene. And the blue color, if you observe the triplet codon ATG1, CAA1, and this CAA, the next overlapped by another genes ATG triplet codon. This is overlapped to the previous ATG, right? And CTG, cytosine, thymine, guanine. So as it's the overlapped genes, what happened during the protein synthesis? This code, this blue color, sorry, green color, code one protein, codes one protein, and let's change the color, green color after the blue color. This blue color, the blue color codes for another protein. The simple one. Let me take one triplet codon, simple. For example, here CAA in first in green color CAA, just triplet. After CA, TGC code for one amino acid. And this overlapped gene actually ATG. If you see the overlapped gene, this is another triplet. For example, this CAA code for one amino acid. Of course, here we can observe the ATG overlapped like this. This is overlapped here. But what I'm saying here, the first triplet CAA, A amino acid. Now, after three completed A, like this, now TGC, thymine, guanine, cytosine, code for B amino acid. B amino acid. Amino acid simple. Remember, don't get forget. And a group of amino acid finally form the protein. Remember it. Now the overlapping gene ATG, which is resemble to the first A, A, T, G. Now this adenine, thymine, guanine, cytosine, the codes for A amino acids or B amino acid? Tell me. Or another amino acid? Of course, it is a overlapping gene, the same nitrogen bases. Now tell me which codes for if previously A or previously B or new amino acid. Hello? Saleha. Nagarani, Navadeep.
Yes, sir. ATG, one amino acid, that's triple.
Of course, right. ATG, one amino acid, but this amino acid resemble to the first amino acid. First amino acid means there where the overlapping occurs, there already the amino acids are formed. CAA, one amino acid. TGC, one amino acid. Is these amino acids similar to those amino acids or not? That means the overlapping gene, what you observed is ATG here, this is called the same amino acid or different amino acid. You did not get my concept. My voice and my slide all both are clear or not, first of all?
It is clear, sir.
Then why are you getting confused? Actually, diagram not clear. That means the images what I download by from the Google, they are not clear. That's why I had to explain like this. So first of all, overlapping clear or not? Overlapping genes, overlapping sequence. Just wait. Already gene structure. But just wait. Let me take ABCD. Instead of nitrogen bases, nucleotide nitrogen bases, for our clear understanding, let me take ABC. A, B, C. Actually, this is one triplet. D, E, F, G. Right, clear? Hello?
Clear, sir.
Now, let's change the color. Overlapping genes C, D, E, F, G, H. This is the overlapping. First of all, overlapping clear or not?
Clear, sir.
Right. Now they are the somewhere another genes. Here the somewhere another genes like that. Here somewhere another genes, right? So actually the red color genes, yeah, I already told you the one triplet. Here three ABC, DEF, GHI, right? Okay. ABC code uh, small a amino acid. DF small b, GHI small c amino acid. Code for example, these are the ABC amino acids. Now this all after transcription A amino acid, B amino acid, C amino acid all together forms a hello?
Protein.
Right, that's enough. Protein. Protein form. Now let's see the overlapping gene transcription. What are the overlapping? Can you read? What are the overlapping genes here?
CDE.
Right, CDE, which are overlapping to the red color CDE, right? These three. And again, FGH,
FGH.
FGH. Of course, they are overlapped. But when we transcription process, the CDE during transcription can code for A amino acid. Hello? This CDE sequence code for A amino acid. Remember this ABC sequence only code for A. Only ABC
A.
code for A. DEF code for B. GHI code for C. Like that. Now tell me CDE code for A or B or C or another new one.
New one.
Right, another new one. It might be, for example, D. Our understanding only. D. What about FGH? Which code for C or B or A? What FGH?
Another amino acid.
Another, it is not A, not B, not
B
C,
not
not
C.
D,
Yes.
not D also, E.
Hmm.
Now the DE together, now DE together, which forms the protein. Proteins. Now how many proteins here formed?
2.
Two proteins. Actually, here the genes, of course, it appear as a two, but not a two. The single gene overlapped. Due to the overlapping, this first gene can produce the protein, and overlapped also can produce the protein.
Protein.
So say one gene, two different proteins are produced. This protein coding gene sometimes overlap. That is the same nucleotide sequence encodes different protein in different reading frame. This is called reading frame A B C D E F C D E F G H. This is called reading frame. Okay,
Okay, sir.
right? This actually this overlapping this mostly found in viruses, bacteria, and in mitochondria also. But now research occurs in the eukaryotes also. Now the emerging research showing eukaryotes, now lately, the overlapping genes tend to encode the protein with higher enteric structural disorder. This type of proteins leads to intrinsic structural disorder than those encoded by the non-overlapping genes. Due to this intrinsic structural disorder, the various diseases are formed in human beings, that means in organisms. So this is the overlapping genes, and another one is the given alternating splicing, and also known as exon shuffling. The clearly given in our textbook, page number 64. Turn on page number 64, diagrammatically clearly given, page number 64, alternative splicing or exon shuffling. That means actually what happened after introns removal, all exons are, let me take pen. All exons are joined. Exon 1, 2, 3, 4. Now the between introns, right? How many exons here in diagram? Hello, in gene, how
4.
many exons?
4.
4. 1, 2, 3, 4. Alternating with introns. Let's change color for our understanding. Introns, red color, introns, red color, introns, red color, introns. Actually, what happened in general process? In general process, only introns are removed and exons rearranged, rejoined. But what happened here? 1, 2, and skip the third one, and fourth joined. This is one process, and sometime what happened? Due to this, what happened? One exon shuffling. That means actually the splicing only in introns. What is splicing? What is splicing? Splicing means removal. Actually, splicing occurs in introns. After transcription process, these introns, radical introns removed. That is called splicing. Apart from the introns, sometimes what happens? Exons also removed. From due to some reasons, exons also removed. So maybe this is one type, or maybe, for example, in this first, okay, sorry. First, second, third, fourth. Exons and red colors introns. 1, 2, 3. Now the skip second one, and third with third and fourth. This also exon shuffling. But anyway, in this exons are splicing. This is called as exon shuffling. This is called exon shuffling. And another important one is gene family. Another important one is gene family. This is very, very important. Gene family and also repetitive DNA. And gene family important for short answer. Let's discuss about gene family. Right. So what is family first? In general definition, what is family? Hello? In general definition, what is family?
Multi genes.
Okay, where the multi genes, how the multi genes are formed? Here the family means, for example, we have the group of genomes, right? How this group of genes comes from? How this group of genes?
DNA.
From which DNA? Where the DNA? Many genes in higher eukaryotic genomes are related to others in the same genome. That means we have the genome, our parents have the genome, our grandfathers have the genome, our ancestors also genome. These all the genomes are similar or dissimilar? Same or not?
Similar, sir.
Similar. So now this is called gene family. That means in gene family, clearly from the ancestors, from the ancestor, for this box, ancestor, offspring, gene. For example, I'm taking one gene. This gene duplicated, duplication process, that means the gene become doubles. The same gene formation. The same gene become doubles and hereditary toward the enter into the another offspring. And again another offspring, another offspring. Like this gene family, the same genes from same genes entered comes from the ancestral genome toward the offspring. Apart from that, apart from that, new genes also formed. New genes also formed. But the new genes, what is the initial gene to formation new gene? The blue color gene formed. Of course, the new color new gene formed. This new gene formed from the duplication from previous one. This previous genes, the part of our ancestor. So this is called the gene family. This is called gene family. Here the gene family given clearly example, one example that is a globin gene family, globin, globin gene family, globin gene family. The globin are the protein components of hemoglobin in mammals. Already you know that. The specialized globins exist that are expressed in embryonic or fetal circulation. These have higher oxygen affinity. Just assume, an embryonic baby directly contact with oxygen. Hello? Can breathe oxygen, can take oxygen directly. The fetal baby or embryonic baby can receive oxygen directly, can receive atmospheric oxygen. Hello? I'm asking very simple question to you. Am I audible? Hello? Hemobindu. Pravina,
Sir.
please
Sir.
respond. I'm asking simple question. That simple question is, fetal baby can directly receive oxygen. That means can receive atmospheric oxygen directly. Hello?
Sir.
Actually, fetal baby inside the womb and they receive the oxygen from the mother, and it has the only globin protein. Globin protein, the character of globin protein in embryonic stage. In embryonic stage, higher oxygen affinity, higher oxygen affinity. But after birth, just simple. Just compare with, for example, this is the mother, blue color, green color baby inside the womb. The mother directly take oxygen from atmosphere, but the baby can receive atmosphere from mother. But where high affinity? In which globin high affinity? Mother or baby? Hello? Hello? Higher oxygen affinity. Higher oxygen affinity. The baby have the higher oxygen affinity than the
Mother.
adult hemoglobin. The serve to capture oxygen at the placenta, moving it from the maternal circulation. So the developing embryo or fetus. But what happened after birth? The familiar mature hemoglobin which consisting to alpha, beta. Alpha and beta. Actually, after birth, the familiar mature hemoglobin 2 alpha and 2 beta subunit. 2 alpha, these are the genes 2 alpha, 2 beta subunits. Replace these the globins. The first actually only globin present, globin gene, only one gene. This one globin gene separated into alpha and beta. One globin gene separated into alpha and beta. And how this alpha and beta comes from? The where this alpha and beta? This alpha and beta from the ancestor. That means mother. Mother gene that shares the alpha and beta genes. Alpha and beta genes. So this alpha and beta genes, the part of the globin family. Part of the globin family. Like this. This is the only comparison from the mother and baby. Like this, if we compare the from our ancestor, the simple assume one thing. Are we the same like our ancestor without any changes? Hello? Hello?
Sir.
Are we same? Are we similar? Are we
No, sir.
similar?
No.
No.
No, sir.
Of course, gene, that means the gene families, what are hereditary passes for generation, there gradually changes occurs, including gene families. That means gene passing genes. For example, our ancestor, the same similar genes is 100%. The same 100 genes are transmitted for generation to generations. But within the 100 genes, now and there, some of the changes are occurs. Some of the changes are occurs. So this is all about the gene family. And another one, next one, repetitive DNA. Repetitive DNA. I will explain tomorrow. Repetitive DNA, moderately repetitive DNA, and higher repetitive DNA. Okay.
Okay, sir.
Right.